Hello everyone, my name is Buonagura, and today we are going to talk about the evolution of multicellularity. It is theorized that the first life forms to exist millions of years ago were all unicellular organisms, meaning that they were single cells with a membrane-bound nucleus that absorbs nutrients from its environment via osmosis and active transport. The question is, how did simple unicellular organisms evolve to form more complex multicellular life? This video is based on a lecture given in the Evos seminar series course at Binghamton University. The lecturer, Rebolleda Gomez, discusses her research into multicellularity using yeast cells to study unicellular and multicellular organisms. Yeast is an excellent organism to study because it reproduces fast, allowing for quick formation of culture. It can exist as a single cell or as a colony of yeast cells. It is safe to work with, and there's already a bunch of background information available because scientists have been working with it for so long. So the experiment begins by taking some yeast cells and placing them in a test tube with nutrients for growth. The yeast cells are the orange circles. The test tube is then incubated for 24 hours, allowing the yeast to reproduce and grow. After growth, the test tube is shaken vigorously or spun in a centrifuge in order to get the larger yeast organisms to settle to the bottom of the tube, as shown by the bolder, larger colony of yeast. The largest yeast are then removed using a pipette and transferred to a new test tube. The process is repeated many times until a considerable culture of large yeast is available. Cells naturally want to grow and obtain nutrients from their environment. The issue, though, is that a cell can grow too large and rupture or perish from malnutrition as the inner volume of the cell exceeds the amount of nutrients that can be transmitted across the surface area of the cellular membrane. One solution yeast have found is to form multicellular units known as snowflake yeast. Snowflake yeast is a single organism is the apoptosis behavior of the cells. Apoptosis is programmed cell death, and in snowflake yeast, specific cells will undergo apoptosis so that the whole can function better. In Dr. Gomez's experiment, multicellular and single-celled yeast were placed on the same media-rich petri dish and allowed to grow for many days. The results showed that the single-celled yeast grew at a much faster rate. This experiment was also performed in an aqueous solution. The results of this experiment are interpreted through figures provided by Dr. Gomez. The first figure shows growth of yeast in aqueous solution to the left and on the petri dish to the right. As you can see, in both scenarios, the single-celled yeast grew at a much faster rate. However, the difference in growth rate is less extreme in the aqueous solution. The second figure shows a more detailed visual of the difference of growth on the petri dish. The first row shows single-celled yeast colony, the second row shows multicellular yeast, and the third row shows nutrient availability. The first image in the second row shows that multicellular yeast initially grow much more quickly than single. However, over the long term, single-cellular growth is happening at a much faster. The difference in growth rates can be explained by mobility and spatial distribution. In the petri dish, the single-celled yeast are spread out more, and the individual cells can obtain all the nutrients they need, whereas the multicellular colonies cannot obtain enough nutrients to grow properly because none of the cells can move in a petri dish. In the aqueous solution, the single-cellular yeast can simply move more quickly than the multicellular and have more access to a larger spread of nutrients and can grow more quickly. Growth curves can be seen to show that single-celled yeast in both cases grow much more rapidly and have a higher potential for growth. Overall, the main conclusions of this lecture are that multicellular organisms can form when desire for growth drives individual organisms to form cohesive units. Resource competition among single multicellular organisms is dictated by mobility and spatial arrangement. And finally, while this concept was not discussed in this video, the need to obtain resources can lead microorganisms to cooperation instead of competition. And different forms of cooperation are very interesting to study to understand more about multicellularity.